This is video number 2, or how to view your DNA sequences in DNA Subway. Before you go into DNA Subway to view your 2 DNA sequences from your seafood samples, it's totally necessary that you actually know each of the vocabulary words that you'll be asked questions on. Those vocab words are right in here in the DNA Analysis Word Glossary. So I'm going to go ahead and go over some words that you'll need to understand prior to entering and viewing your DNA sequences in DNA Subway. The main words that you'll need to understand are electropherogram, number of N, your Phred score, and last, the peaks and background noise. Each of these terms I'll be referencing while looking at an example of a DNA sequence in DNA Subway. Make sure that you revisit this DNA word glossary so you understand the definition of these terms when you're looking at your own DNA sequences. So we'll go ahead now and go back to DNA Subway. This is what DNA Subway looks like after you log in. To view your 2 DNA sequences, you want to click on what's called the Sequence Viewer. What will open up are your 2 samples sequences. You'll see over here on the left that I'm showing an example from Section A, Team 2, Sample 1, and Section A, Team 2, Sample number 2. To view your sequences, you go ahead and you'll click over on the blue link to open up what's called an electropherogram. This is where it's super important to understand the terms that I just pointed out to you in the DNA word glossary, which is in step 7. You can see up at the top, this row is your actual DNA sequence from your seafood sample. If you use the cursor, you can scroll all the way to the right to view your entire DNA sequence. Note that red A's represent adenine, blue C's represent cytosine, red T's represent thymine, and then you have black G's or guanine. Scrolling to the right, you can see your entire DNA sequence. Looking at this DNA sequence, you can tell that it is around 690 base pairs long, meaning it has about 690 bases, or letters A, C, T, and G. This first row is your DNA sequence. This second row, where you see a light blue horizontal line, and you see the bars, is called your Phred score. If you scroll all the way to the right, you'll see that there's a Phred score associated with each base. Underneath this C, you'll see this Phred score. Underneath this T, you'll see this Phred score. In order to understand what the Phred score is, make sure to read about it in the DNA word glossary. Down below, the last thing that electropherogram points out and shows is what is called the peaks and background noise. All these little peaks that you see at the very bottom of this electropharogram show the quality of your particular DNA sequence. Peaks and background noise is another vocabulary term that you should learn about in the DNA word glossary. To view your other DNA sequence, you can click on this other sample. In this case, A21 looks much different than A22. A22 is actually a sequence with somewhat good quality. You can tell that by scrolling over and seeing that you have a complete DNA sequence. A21, however, looks very different. A21 is an example of a DNA sequence that has very low quality. The next video, in step 7, we'll talk about what to do if one of your samples are low quality. Right now, click out of video number 2 and open video number 3 if one of your samples or sequences are low quality, meaning that you have a sequence that might look something like